Hello and good afternoon. My name is Posy Busby and I'm speaking to you today from Oregon State University, where I'm an assistant professor. And today I'm going to talk to you about research in my lab addressing the assembly and function of the leaf microbiome. Before I get started, I just want to thank Gary and Eric for the invitation to speak today. I'm really excited to be a part of this symposium, and I'm looking forward to the discussions that we have this afternoon. Okay, so for many of us, it's springtime. And what that means is the reemergence of leaves in all sorts of shapes and sizes. What you're looking at here are leaves from the northern forest. But despite this variation in form and function, one thing that's common to all of these leaves is they're all colonized by microbes. What you're looking at here are bacteria on the leaf surface, and in this middle panel, you see bacteria that are not only on the surface of the leaf, but they're actually beginning to colonize the interior portions of the leaf via the stomata. Now today I'm going to talk about leaf fungi. This is the group of organisms that I study in my lab. Now, leaf fungi form a taxonomically diverse group. They include members both from Ascomycota and Basidiomycota, the two major groups of fungi. So you're looking at individual leaf fungi here that were isolated from leaves. Now, in addition to their taxonomic diversity, leaf fungi are also functionally diverse. So some leaf fungi are pathogens. They cause disease in their host plants. But living side by side, pathogens are another group of fungi called endophytes. Now, endophytes live cryptically in leaves, so they show no outward sign of their presence. So you can look at these asymptomatic leaves and you might have no idea that living within these leaves are endophytes. But when you bring the leaves into the lab, you can culture out endophytes from these leaves. And once back in the lab, you can then conduct inoculation experiments to test the ecological function of these fungi in their host plant. Now, in these experiments, we found that many endophytes are commensal symbionts. They have no appreciable effect on their host plants, but others are capable of modifying plant disease severity. And this is an emerging view of an important function of leaf endophytes. So this was something that I showed in a recent experiment where I collected and tested over 30 different individual endophytes of the black cottonwood, which is the cottonwood that's native here to the Pacific Northwest. These were common things like Epicoccum, Cladosporium, Alternaria, Trichoderma, common in cottonwood, but also common across many other, or common as endophytes in many other plants. And I tested them individually, inoculating an endophyte and then the pathogen, the tree's major leaf pathogen, which is a rust pathogen, and asking whether the presence of that endophyte influences the outcome of disease, compared to a control group which received no endophyte, but just the pathogen. And here's what I found. Isolates of over 5 of the 7 fungal genera that I tested were capable of modifying disease severity, in many cases decreasing disease severity, but in one case increasing disease severity. So again, this suggests that disease modification is a common ecological function of leaf endophytes. But this study, and really most studies of leaf endophytes, focus on endophytes individually and test their ecological functions individually. And this is so despite the fact that leaf fungi don't occur singly. They form in complex communities. So what you're looking at here is actually a leaf imprint on rich media on this Petri dish here. You can see all sorts of different fungi that are growing out from the leaf onto the media. Now, because leaf fungi have differential effects on disease severity, and because they form complex communities in wild plants, that I would argue that we need to ask the question, to understand ecological function, we need to ask the question of how does this fungal community assemble into its composition, and then what is the consequence of the community as a whole for ecological function. So research in my lab has been addressing this question by testing the hypothesis that host genotype and the environment together impact host microbiome composition and function. Now, in essence, we're treating the leaf microbiome as an extended phenotype of the host, both its composition and its function. So we're really borrowing approaches from evolutionary ecology to understand the relationship between plant genotype and phenotype. Again, phenotype being the extended, the microbiome as an extended phenotype of the host. So today I'm going to talk about 2 experiments testing this hypothesis. One was conducted in a common garden, and the second was conducted, was a more controlled experiment conducted in a greenhouse. Now, both of these experiments were conducted using black cottonwood, the tree I already mentioned, where I collected and tested over 30 different commonly occurring endophytes and showed that they're capable of modifying, or that many of them are capable of modifying severity of the tree's leaf rust, which is shown here. This is also a wonderful study system, in particular for addressing host genetic and environmental effects on leaf microbiome phenotypes because of the incredible genomic resources that are available for this tree. So Populus trichocarpa was the first tree to have its genome sequenced. And since then, over 1,000 trees have been resequenced. You can see these trees are coming from the trees that have been resequenced come from wild tree populations. This resequencing effort has yielded a SNP library of over 28 million SNPs, so we can associate genotypic variation with phenotypic variation. Moreover, another amazing feature of this system is that these over 1,000 genotypes are planted in multiple common gardens throughout the Pacific Northwest, enabling common garden studies. So the first experiment I'm going to talk about today, again, is a common garden study testing host genetic and environmental effects on microbiome composition. So in this experiment, we control host genotype by planting the same set of genotypes in multiple locations. We control the common garden by planting all the genotypes in different and distinct gardens, but we allow fungi to infect these plants naturally. So we rely on the natural infection process. The two gardens that I'm going to talk about today, one is in Corvallis, where Oregon State is located. This is a cool, wet environment. Second is in Boardman, Oregon, and Eastern Oregon, which is a hot, dry environment. So contrasting environments, climatic conditions are expected to influence fungi as our host genetic factors. For 2 years, we sampled leaves from these gardens, and also both early and late in the season. What we do is we bring those leaves back to the lab, and then we extract DNA from the leaf tissues, and we amplify the ITS region, which is the barcode region for fungi. Once we get that sequence data back from individual leaves, we can compare the ITS sequence data to published ITS databases to identify which fungi are in the leaves. Now, in this effort, we found over 1,000 different fungi across these two gardens and in early and the late season, and that's a lot to think about and to try to visualize. So, what I'm showing you here is an ordination analysis where we compress the communities into 2 dimensions. Now, each data point in this ordination analysis represents the fungal community of an individual leaf. Data points closer together in space are more similar. Data points farther apart are more dissimilar. The purple colors are the leaves, the fungal communities from Corvallis, and the orange are from Boardman. So what you see right away, and I should also say the lighter colors are early season, the darker colors are late season. So what you see right away is clustering of fungal communities within each of these gardens, suggesting that within the gardens we see similar fungal communities, although there are differences early, from early to late in the season. But we see that the communities in the 2 different gardens are quite dissimilar from each other, separated in ordination space. And indeed, this has been shown in several other systems with plant-associated microbiomes that environment very strongly influences the composition of plant-associated microbial communities. What we wanted to do next was to dig into the communities and ask how important is host genetic variation for these communities. So to do this, we conducted first a principal component analysis on the SNP data, the genomic data, and then we used the top 10 PC axes as predictors in our models of community dissimilarity. And these axes explained about half the variation in the SNP data. When we do that, we see that host genetic variation explains about 5 to 8% of the variation in the Corvallis garden, but did not explain a significant amount of variation in the Boardman garden. So right away, we're seeing an interesting genotype by environment interaction, where genetic variation is important for community composition in the cool wet garden, but not in the hot dry garden. And our ongoing efforts seek to try to understand and unpack that genotype by environment interaction by looking at how fungal biogeography and leaf trait plasticity and other factors might contribute to these differences. Another really interesting thing that we saw in this experiment was that the location within the common garden, the spatial location of trees in the common garden was important for the composition of the community, explaining between 10 and 15% of the variation. This to me suggests that stochastic variation in species arrival order could be important for composition. And that's something that I'll talk about in the second part of that talk, in the second part of my talk. So kind of digging into this genetic effect here in Corvallis, we wanted to know which taxa in particular could be driving this relationship between host and endophyte communities. So next what we did was to calculate the heritability of individual fungal taxa in the community. And we did that for 3 different data sets in Corvallis where we see this genetic effect, the 2016 data and the 2 2017 data sets early and late season. So for each fungus that's arrayed in this phylogenetic tree, these are the common fungi in Corvallis, you'll see there are 3 data points representing these 3 data sets. The shade of blue represents the narrow-sense heritability value from 0 to 1, the darker colors indicating a higher heritability. And if there's a red ring around it, that indicates that it's significant, statistically significant after comparing for multiple comparisons. So what we found here first was something that wasn't terribly surprising. You see that there's a genetic basis for plant pathogen phenotypes based on the abundance of these organisms in the sequence data set. This isn't surprising because we know there's a long history of research on studying the genetic basis of resistance to pathogens. And so this comes as no surprise. We see Melampsera, this is the leaf rust that was tested experimentally, pops out Tophrina, Venturiae, and other pathogens. But what was more surprising is we see that there may be a genetic basis for these endophyte phenotypes. Many endophytes also emerge as significant in this analysis. Some of these endophytes are pathogen antagonists, so they do have known ecological functions, but most of these have no known ecological function, suggesting though that there could be an important ecological function that needs to be further studied, and suggesting the possibility that there's even a genetic basis to this interaction between plant and endophyte here. But of course, we have to be careful with this type of interpretation because this is a field study, and so there's uncontrolled infection and assembly in this community, and the community includes pathogens. So these putative genetic effects on endophytes could in fact be genetic effects on pathogens. Could truly be genetic effects on pathogens, and pathogens then could be influencing the abundance of endophytes in the community. So it could just look like a genetic effect on an endophyte. So to more specifically look at the relationship between a plant genotype and its endophyte community, what we need to do is to move into the greenhouse environment where we can control for not only host genotype, but we can also control for the endophyte assembly process. And here, what we really wanted to do was to follow up on that finding that spatial location in the common garden mattered for community composition. And so what we're doing here in this greenhouse study is we can actually vary the arrival order of members, of the individual members into the community to evaluate whether that is the case. So just here is a little bit of a schematic to help you get a sense of the design of this experiment in the greenhouse. So in this experiment, host genotype was controlled. We used both Eastern and replicates of both Eastern and Western ecotypes. And then of course, as I mentioned, we also control the endophyte community by using a synthetic community of 8 members. Now in this 8-member community, we have different assembly histories with 1 of 5 different members of the community arriving first. And then for each of these different histories, after the initial colonist arrives, 2 weeks later, the rest of the community arrives. Now, about a month later, we used the same ITS metabarcoding to characterize the composition of these communities. And then after that, we inoculated the plants with rust to evaluate how assembly history and host genotype can individually and interactively influence the outcome of disease in this system. So what did we find? We first found support for the fact that host genotype indeed does influence the composition of endophyte communities. And again, this is with a synthetic community in the absence of a pathogen. So here, individual data points represent the fungal community found on individual genotypes summed across the different immigration histories. So separation in space indicates differences between one genotype to the next in the composition of the community. We also see clustering of Eastern versus Western genotypes, suggesting that traits underlying these ecotypes could be important for host genetic effects on the composition of the community. Digging deeper into these communities, we can identify how host genotype and immigration history influence the abundance of individual members of the community, of the 8 members of the community shown here. And this is a heat map showing standardized relative abundance of each member of the community across the different immigration histories, which are shown along the top. And then for each of the different genotypes, which are color-coded here by their ecotype. So first, overall, we found that 7 of the 8 members of the community were sensitive to host genotype. Again, confirming this relationship between plant genotype and endophyte abundance. And we also found that 7 of the 8 were sensitive to immigration history, again, confirming the importance of stochastic variation in arrival order for these organisms. Just to show you one example, you can look at here at alternaria, where first I should mention that the brown colors indicate greater standardized relative abundance. So when you look at alternaria and you look across all of these different immigration histories, you see that they're browner colors, so greater standardized relative abundance when alternaria arrives first. So it benefits from early arrival. You also see there's an ecotype effect. We see greater relative abundances on the eastern compared to the western. And then within the western, though, we can see there's variation from genotype to genotype, with some having greater relative abundances. So next we wanted to know, how do these effects on individual taxa and the community overall, how do they influence the resulting disease severity once we inoculate with rust? So what are the consequences of this assembly history for plant disease? So to answer this question, we first separated the genotypes into those that were resistant to rust disease and those that were susceptible. And that's because the resistant genotypes expressed major gene resistance to the rust, which really shut down rust development. And so in these genotypes, we don't see that endophytes play a role in modifying disease severity. But it's where it's in the genotypes that are susceptible to the rust where we see interesting interactions taking place. So on the y-axis here, we have disease severity. The color code, the different colors represent different immigration histories. The panels are different genotypes, and the dotted lines are the mean, are the control. Represents the control group for each genotype. So this would be the level of disease that develops on plants that were inoculated with just rust, no endophyte. So first we see that here, endophyte communities, and again, this is communities, not individual endophytes, can modify disease severity and primarily reduce disease severity. And they reduce disease severity in this experiment. But this didn't happen on all genotypes. We see on some genotypes, we see disease reduction, but on others, we see no effect of the endophyte community. So it's a community by genotype interaction. Next, we also see evidence that immigration history is important for disease outcomes, but we also see that this also varies by genotype. And for an example, we can look at Ariobasidium in orange here, where we see in East 1 in this genotype, Ariobasidium has no effect. When this member arrives early, we see that there's no effect on disease outcomes, but in West 5 and in East 2, if Ariobasidium arrives first, we see a disease reduction. Now, I expected that these differences, these genotype by immigration history, interaction effects on disease severity could be explained by the relative abundances of different members of the communities that we see that are sensitive to these two factors and to the composition of the composition of the community overall. But in fact, we didn't see evidence that the composition of the community explained these outcomes. And what that suggests is that microbiome composition and function or disease modification was decoupled in this experiment. So composition did not predict function. And that implicates other factors in disease modification by endophyte communities. There could be higher order interactions in the microbial community that are important. Endophytes could potentially be modifying the host defense response, immunity, differentially among these genotypes. So these are things that we need to further explore. But importantly, I want to point out that this decoupling really emphasizes the need to understand the assembly process in the community to fully understand function. We cannot simply predict function from composition. All right, so to summarize, I showed you that individual fungal endophytes and endophyte communities can modify plant disease severity. So these effects are really neither good nor bad. They are complicated. It really will depend on the composition of the community and the assembly of the community. Second, a variety of host genetic and environmental factors were important for influencing the assembly and function of the community. And here I would argue that we're really just at the tip of the iceberg in terms of understanding how host and the environment influence the leaf microbiome. But again, one thing that we have learned and that has emerged is that the composition and the function can be decoupled. Finally, I would argue that improving our understanding of plant endophyte symbiosis not only improves our understanding of the ecology and evolution of this fascinating relationship, but I think it can also potentially improve crop health via microbiome management in agriculture. I didn't talk about this today, but I think this is an important application of this work. With that, I want to acknowledge my lab, in particular, Devin Leopold, who led most of the work that I talked about today, Jacobson Lab as well, as a collaborator on this work. And I'll also thank my funding sources, and I look forward to taking questions. Thank you.